Shoma, reply.
It's not in presentation mode yet.
No.
It's a slide mode.
Yes.
Okay, I'm gonna stop sharing,
Yeah, do it again, please.
and
Ron.
then I will go back into my presentation.
Oh my God. Sorry.
Okay, Shubho, my video and audio is okay because I'm not using a laptop today.
It's okay.
Share. Okay. Now, you can see it, but I've got to get it into slide mode, eh?
Not quite.
Uh,
This is not yet in slide mode, Ron.
Okay, how do I get it into slide mode?
If you click here in the presentation mode, this one, did you click already or it's not working your side?
Is
Yeah.
it
Yeah, yes.
Yeah.
Good.
Good.
now? Okay.
Yeah.
And then I want to get my screen pointer. Okay.
All good.
Okay, is that going to be all right?
Yes.
yes, yes.
Okay.
So, Shoma, are you ready?
Yes.
Okay. So now I'm going to start recording and okay. Thanks everybody for joining today's webinar. This is Ron's second webinar. It's also because his experience is so vast, we can't cover it in one webinar. Maybe more, 3-4 webinars is very few for his experience, but we are glad. Today we also again planned for this webinar, and as you know, that certificate link will be provided at the end of the webinar, so please don't ask multiple times. And those are registered for this webinar and present in live session and submitted their feedback, definitely they got the certificate after 3-4 days. Unfortunately, our website is today is down for the maintenance, but it will work in a few hours. So don't worry, we can communicate with us at admin.bioengine.com. Today's I have Soma and Dr. Priyanka Gupta to help me this webinar session. Soma will introduce speakers and Bioingene, and Priyanka will help me out for the discussion and question answer round. Put your all questions in YouTube chat box. I will collect them further on. And we already planned few questions accordingly. Previous webinar, experience from the previous webinar, and today's in respect of today's topic. So, Shoma, please start.
Hello and welcome to BioEngine webinar number 68. BioEngine is a non-profit organization created to promote plant science research worldwide. This is an open platform from which plant scientists can present their current advancements of their research to the world. We hope future scientists can gain perspective and inspiration watching the esteemed plant researchers talk about their scientific accomplishments and views on the building of future of plant science. We are thankful to the speakers and viewers of our Bioengine family for your interest in the webinar series and for your appreciation of our efforts. You can register for Bioengine webinars through our website, bioengine.com, or our social media page. You are live viewing this webinar and you can watch it again later on the Bioengine YouTube channel. So please remember to subscribe so that you don't miss out on any of our events. After attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided in the YouTube chat after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is Advancing Genetic Gain Through Rapid Variety Adoption by Dr. Ron DePauw, Science Advisor, SeCan, Canada. Let me give a brief introduction of Dr. Ron for our new viewers. Dr. Ron is an internationally recognized scientist. He has devoted his life for the betterment of society and the farm economy, including resilience to climate change. Ron was a member of the Independent Steering Committee to the Global Wheat Program of the Consultative Group on International Agricultural Research managed by CIMMYT. He serves on the Borlaug Training Foundation board, which collaborates with CIMMYT to train the next generation of hunger fighters. Ron has actively participated in the registration of 70 wheat and 6 triticale cultivars for the past 23 years. 40% to 60% of all wheat grown in Canada were derived from cultivars that he and the Swift Current Research and Development Center team developed. Cultivars developed by Ron and his team exemplify the use of environmentally friendly and low input cost genetic resistance to control a multitude of diseases and insects. Ron worked for Agriculture and Agri-Food Canada for 42 productive years, excelling through leadership and innovation in the wheat industry. He has received numerous awards, including the Order of Canada, Saskatchewan Order of Merit, Queen Elizabeth II Platinum Jubilee, Gold Medal from the Professional Institute of Canada, which has only been awarded 7 times to someone in agriculture, and an Honorary Doctor of Science from the University of Saskatchewan. Dr. Ron, welcome back to our show. The BioEngine platform is all yours now.
Thank you very much, Soma. Yes, thank you very much. And greetings to everybody that is listening online from all of these diverse places around the world. So yes, it gives me a great deal of pleasure to share with you about advancing genetic gain rapidly to get it into the farmer's hands, get it into the farmer fields. Because if you're making something innovative and it's not used, it is really an unfortunate waste of resources. So in my presentation today, I'll be speaking about the quality attributes of Canadian market classes. In Canada, we don't think of wheat as wheat. We think of wheat as being packaged into various kinds of market classes to make various kinds of food products. And that will affect the relative size of the market. Then I'll do a fast review of a flowchart of variety development and then that initial Pedigreed seed production. And how do we get that seed, that innovative new breeder seed from a breeder to a seed company? And from there, the production of the Pedigreed seed to multiply it, play it up, promote it, and distribute it. And of course, then what is the role of farmer to farmer in variety adoption? And when I'm speaking about farmer to farmer, I'm thinking of the professional seed grower becoming aware of the genetics that he's multiplying and then informing other farmers of it. And of course, we're respecting intellectual property rights in that regard. And I'm going to end with the of these genetic gains being delivered to farmers. Do we have any proof of that? So as I was saying, in Canada, wheat is not wheat. We think of wheat being in market classes to make end products. So if you think of you're looking for confectionery products, whether they're cookies, cakes, crackers, that kind of a product, then the quality attributes is to have a soft kernel, a low protein quantity and a weak gluten. And of course, in the Canadian classes, we have the Canada Western Soft White Spring, the Canada Eastern Soft Red Winter, and the Canada Eastern Soft White Winter. But if you're going to be making other kinds of products, whether they're French, Brazilian, Arabic bread, steam breads, noodles, chapatis, filler, and wheat, but many, many, many of those sorts of products, well, then a medium hard kernel with a medium quantity of protein and a medium gluten strength would be used. And that would be the Canadian classes of the Canada Prairie Spring Red, a Canada Western Red Winter, or a Canada Eastern Hard Red Spring. And carrying on, if you're looking for certain kinds of pan breads and noodles, then you'll be looking for a hard kernel, medium protein content, but on the type, depending on the type of the noodle, if it's a medium strong gluten, then you'll want a Canada Western Red Spring at 11.5 protein. But if you're wanting a strong gluten, then you'd go to the Canada Western Red Spring at 12.5 protein. And if you're looking for end products where you're wanting the wheat to be blending for blending purposes to carry weaker gluten materials or certain kinds of pan breads and certain kinds of noodles, then you'd want a hard kernel, high protein, and a strong gluten. And then you'd be in the Canada Western Red Spring at 13.5 protein. And if you're making pasta or couscous, bulgur, then you want a very hard kernel, a medium protein quantity, and a strong gluten, and that would be the Canada Western Amber Durum. And so all of these different classes of wheat don't have exactly the same value because the market for, let's say, confectionery products might only be 10% of all of the wheat that is grown in a particular country. So then the size of the market class reflects its approximate value. So this big class, the Canada Western Red Spring, is the high protein, hard kernel, and medium to strong gluten, depending on the protein segregation. And Amber Durum is the other very big class, and Amber Durum is actually the most expensive class in the world, and the hard red spring wheats is in a class of the premium wheats around the world, like the Australian Prime Hard and the US Dark Northern Spring. So you can see the other classes are much smaller. They make up maybe 10% of that total production. So when one is building a variety, one has to think about, well, what is the quality package that I'm building in? If I'm building in a new trait for a new disease resistance or a climate smart trait, and I put it into a minor class, then the opportunity for having an impact at scale in farmer fields is much reduced. And of course, when it comes to variety adoption, the multiplication of the seed, when the seed companies know what is going to be the market eligibility, then they have a very good idea about how to scale up the variety to meet that demand. So looking at the average of the crop insured acres for the last 5 years, you can see that hard red spring wheats is over 5 million, Amber Durum is 1.7, and the others are all small, out of a total Western Canadian crop of 7.6 million crop insured hectares. And crop insurance covers maybe 85% of the area. The other 15 to 20% that is not insured, farmers are growing them completely at their own risk. Timelines to deliver a new cultivar to producers. Well, the first thing that's required is to develop that inbred line. And that takes about 4 to 5 years, depending on the strategies. And then, of course, one has to demonstrate that this inbred experimental line that the breeder's in love with will, in fact, bring value for cultivation and use. And that determination is done by the Prairie Recommending Committee for Wheat, Rye, and Triticale evaluation teams of agronomy, disease, and quality. If the experimental line shows value for cultivation, it'll be registered, or support for registration will be provided by this Prairie Recommending Committee for Wheat, Rye, and Triticale, and the developer can then get the variety registered for cultivation by the Canadian Food Inspection Agency. And something that's unique to Canada and extremely valuable when it comes to rapid adoption of varieties is the simultaneous determination of the market classification eligibility. And that's done by the Canadian Grain Commission. And this applies only to wheat. It does not apply to malting barley and milling oats. So it's something quite unique to wheat, and it has a very big implication because once the new variety is supported for registration, we know what its agronomics are, we know what its disease response are, and we know what market classification eligibility it has. Then we can engage in seed multiplication, promotion, and distribution by seed companies. And they will scale it up to meet what is that expected size of the market. And the Pedigreed seed certification is done by the Canadian Food Inspection Agency and the Canadian Seed Growers Association. So then in total, the time to get that new technology that's developed into farmer fields would be from 11 to 13 years. I'll just do a fast review of a schematic of a breeding program, cultivar registration, and seed multiplication. So first of all, do the crossing, whether it's biparental or top cross or one back cross. And then you go through the generations of inbreeding. But you can accelerate generation advancement by using a contra-season nursery for inbreeding, selecting, and multiplying. And there's lots of questions about speed breeding. Well, this is a form of speed breeding. Anytime you can get more than one generation per year, you are on your way to speed breeding. And in this particular schematic that was used at the Swift Current Research Station of Agriculture and Agri-Food Canada, the contra-season was used for not only inbreeding, but also selecting and multiplying seed so that you could conduct early generation yield testing starting in F4 for diseases, agronomics, and grain quality. So the breeder is building in grain quality attributes. The breeder is targeting one of those market classes right from the get-go. And I mean get-go. The crosses, the strategic crosses that would be made would be addressing whatever the target is. If it's, climate change, and you have to build in all of the disease resistance, that strategic cross has to be made there, also with consideration to what quality market class are you targeting. So the parents have to have all of that big bag of genes, and because you've got a big bag of genes segregating, you need a very large F2 population, very large. Like 10,000 plants is a minimum for a cross that is segregating with so many genes. And then after an inbred line has been developed and it looks very promising, it'll go into these registration trials. To enter into the registration trials, which is a 3-year process, and get support to register. If getting support to register. If one is using doubled haploids, again, you've got to make your cross, you've got to produce the DH, and of course, then you've got to do the selection and multiplication, moving into the registration trials. And so the big thing with trying to accelerate adoption into farmer fields, then this step here of initiating breeder seed concurrently with the evaluation that is ongoing in the registration trials is very important. This can save reduce the time to get into farmer fields by 3 years if one is practicing concurrent development of breeder seed at the time of going through the registration trials. So if the breeder gets support to register, then the next step is going to be commercialization. And the commercialization then involves the release of the breeder seed to a selected seed company to go on to multiply the seed, to blow it up for a commercial launch. And that usually takes about 3 years. So in terms of, just to give you an idea that the variety supported for registration and commercialization, there's lots of varieties that gets, and I'm just looking at 2 market classes, the hard red spring wheat and the amber durum class. So supported for registration by year, you can see there are a number of varieties in the last 12 years that have been supported for registration, a total of 93. But note here, not all of them were commercialized. 20 of those 93 were not commercialized. And in amber durum, it was the same thing. You get lots of varieties that are supported for registration, but note, 10 of that 40 were not commercialized. And the reason for that is farmers want better. They want better varieties, not more Me Too varieties. So even though these varieties, these 20 varieties were supported for registration, when it came to the assessment of will they be really a truly better variety in farmer fields, 20 of those 93, even though they were supported for registration, it was decided no, they don't have enough possible attributes to make it onto farmer fields successfully. So I'm just going to do a fast look at how is the certified seed now made available, going to be generated, going to be produced. How are you going to preserve the genetic identity and the purity of that seed as you multiply it up? So this is a schematic of how it is done. The first step is about variety development and the production of that basic seed. So this is the breeder's job, 1 and 2. And then steps 3, 4, 5, 6, 7, 8, 9, 10, and 13 is done by seed growers. These are specialized seed farmers that are trained in the production and retaining the identity and the purity of the seed, and for scaling it up. So I'm going to now, I'll deal with that a little bit more. But the first thing is about the production of that basic seed. And with the concurrent progression through the registration trials by the breeder. And in year 1, the breeder will have some single plants. From the single plants, they'll go to plant progeny rows. And in the 3rd year, it'll be multiplied from the single rows to pre-breeder rows to make the breeder seed. And at this point, he might have 300, well, then you have where from 200 to 400 or even more breeder seed. And that breeder seed then is turned over to the seed company that was selected based on a request for proposals to produce the select seed or foundation seed from that breeder seed. And then the next step is to multiply that seed to foundation and registered, and then in year 6, registered and certified. And launch can be at the end of that season, so that in year 7, it is in farmer fields. The upper right is an example of a single row per breeder line. And the breeder will assess, are these lines similar from one to the other? And are they uniform within? And if they are not uniform within, they will be discarded. And if a whole row is not looking like another row, then the whole row is discarded. Another way that it is done, if is to have 2 rows per breeder line. And again, the same steps. This is a pair, this is one breeder line that has 2 rows. And does it look similar to the others that are there? And if not, it gets discarded. And this is an example of where there are 3 rows per breeder line. So this is breeder line 1, row 1, breeder line 1, row 2, breeder line 1, row 3. And again, it is an assessment of, are these 3 rows similar to the other 3 rows of the other breeder line, and so on and so forth. And what is the variation within? And it's always an eye to making all of these breeder lines looking similar. Here is an example of where there were 4 rows per breeder line. So these 4 rows are all part of one breeder line. And the breeder decided this 4 rows of this breeder line was atypical, did not look like the others. So it was removed. You can see back there where these gaps are. And that is the job of the breeder and his team. So then public breeding institutions tend to release their varieties using a request for proposals. And that proposal will include information about the variety that has been registered, its attributes for agronomics and disease, as well as what is its market classification. So the request for proposals then is made available, or it's offered to seed companies to bid, to make a proposal on that new variety. And I'm using examples here for some criteria, examples of criteria to assess proposals in 2024 that from Agriculture and Agri-Food Canada. So the first criteria was the company profile. Well, who are you as a company? And that gets you 10% out of 100. And then what is your marketing and production strategy? How are you assessing the size of the market for this particular new variety? How do you plan to promote this variety? How do you plan to promote it so that making other farmers, other commercial farmers, aware of this new variety? And what are you thinking about sales and royalties returned? And a very big component is, what will you do to protect intellectual property? How will you protect it? And then the final financial offer is 35%. That's like, what is going to be your upfront bid? And what has been your investment in research in the last 2 years? Are you a supporter of variety development? Are you a supporter of agronomic research? Are you a supporter of disease resistance research, etc.? So then for a company, when they're looking at marketing and production strategies, well, there's things that they can do by way of entering the variety, supporting the variety into provincial regional performance trials. And from there, with that information, to help to follow it up, to be sure it's put into provincial seed guides. And what are there by way of field days and promotional events that you can be, as a company, supportive of, to make it known? Like this Ag in Motion is a very large Western Canadian summertime show where commercial farmers will come in the busloads to see not only the plant varieties, but also equipment and new technologies. And what are you going to do about demonstration trials and field days and billboards and digital ads and print ads? And are there opportunities for co-promotional materials, contra-season seed multiplication? Like, not everybody is willing to take this basic seed, to another country in the southern hemisphere and multiply it. But by doing that, you can have a major impact on the quantity of seed that you have available for commercial release. So when it comes to seed companies, there's small companies owned by farmers such as Cantera Seeds, FP Genetics, Penwest, SeCan, which is a not-for-profit, non-exclusive, open. The membership is, nominal. Seed Depot and SeedNet. And then you have medium-sized companies, and they're more like acting like an umbrella for small independent seed companies. Like you have Alliance Seeds, Proven Seeds, Ag Solutions, and Richardson International. So there's a number of seed companies. And interestingly, none of these seed companies develop their own varieties. They are obtaining their genetics from public and private breeding institutions based on request for proposals. So what is SeCan? Well, SeCan is has more than 600 independent Canadian seed businesses engaged in the seed production, processing, and marketing. And they are really owned and operated by family farms. So SeCan is Canada's seed partner and actively seeks to promote success in Canadian agriculture. And SeCan is the largest supplier of certified seed in Canada, with more than 400 varieties in 40 crop types. And SeCan members are major supporters of seed variety development, having returned over $150 million in royalties and research funding since it started in 1976. So in many ways, we hear a lot of talk about farmer-to-farmer organizations in various jurisdictions around the world. And I'd like to point out that SeCan is a type of farmer-to-farmer organization operating under membership rules and respecting intellectual property rights. So the next step is going to be about examples of multiplication, promotion, and distribution. But going to the flowchart, we are now, we got the seed, we got the breeder seed, it's been developed. We are now with the seed growers, operating under, let's say if it's a SeCan variety, the SeCan will be overseeing the seed multiplication. So they'll be selecting growers for doing that multiplication of that basic seed of the breeder seed that has just been received to produce the select and foundation seed. Work with the growers that are specialized in field, prior field considerations, the seeding equipment, sanitation, the isolation requirements, the roguing, being sure that the field gets inspected by an independent auditor for certification processes. There's going to be the harvesting equipment, sanitation, the harvesting, the storage, the cleaning, and then of course there is the step of seed testing for purity, germination, and that is done by independent labs. And if all of those criteria are met, then the Canadian Seed Growers Association, authorized by CFIA, will then bag it. So that would be year one of that little bit of 300 kilograms of breeder seed to produce selector foundation. Then it goes back up here all over again to do the produce the foundation and or registered, and back in here, and then the third year loop will go on as well, and that remains as a closed loop to ensure the production of the genetic purity and retaining the genetic identity without getting it stuffed up and mixed up in any of that kind of way, so that you have a large quantity of seed at the end of year 3 to do a commercial launch to make it available to commercial growers who then will sell their product into the food processing sector. So some examples of seed multiplication and commercial launch. So here we are in year 1. We have the seed we have to plant in tons. And then I have a multiplication rate and production. So we have 300 kilograms. That's all there is. And if it's a low multiplication rate of say 25 to 1 or high 65 to 1, then this is the production in tons. And this production in tons is used then to go into year 2. And again, depending on the multiplication rate, your harvested production will be that. In year 3, and this is what you have to enter year 3 with, your tons with a multiplication rate, so that you can see that at the end of year 3 for your commercial launch, you could have somewhere less than 5,000 tons to 27,000 tons to 82,000 tons of seed for your commercial launch. So that's a lot of seed to now be selling, possibly selling, depending on what you think is the market size. So the commercial launch then, this is what the farmers in year 4 will be doing. And if they're planting at, say, 90 kilograms per hectare, well, then it would be, say, about 52,000 hectares of land planted, or 330 hectares, 1,000. And this would be over 900,000 hectares. So you can see that it's possible to have a major impact depending on how you scale up the variety and make it known to generate for year 4. This is demand pull by the commercial producers. So making this new genetics known is very important. So what is the role of the seed grower in variety adoption? Well, the seed grower is a specialized farmer. You have to know how to produce that genetics and have a quality assurance manual for the production of that seed. So the seed grower is really like a specialized farmer who is a local influencer. And generally, the seed grower is a family farm owner-operated business. And the seed grower knows the variety because she or he grows it. They're a local leader in the community. They're knowledgeable and trusted. And the seed and information is close to the customer. And the seed grower will communicate not only in person, but their various networks and using social media. The role of the seed grower plays a role in intellectual property protection. They have to inform the farmer if the variety is protected by UPOF 91, and has to then inform the farmers about what does this mean? What are the rights of the farmer? What are the rights of the developer? So the seed grower is involved in the support and education of intellectual property rights as well. So here are just some examples of promotional activity where this would be SeCan promoting some new varieties that they have gotten marketing rights to. And this would be like the SeCan reps that would be at an Ag in Motion show where there'll be hundreds, thousands of farmers coming. And so this is a great way, but it takes a great deal of planning to plant these display plots of your new genetics, and then to have your staff trained and informed to inform the farmers. And as I was saying, it's a one-stop shop. You know, all of the equipment manufacturers will be there with their newest and best equipment. So it's a great show. Another thing that gets to be done by in this case by Trent Whitting, a SeCan marketing rep, is to put out demonstration plots. And he'll have all of these different varieties that SeCan is multiplying. But the one strip would be normal nutrients, and the other strip would be high nutrients. So he's able then to demonstrate what is the relative straw strength of these varieties. And this happens to be a drone shot. And you can see over here in the upper right that this variety is quite weak strawed compared to the ones on either side of it or in the background. And here in the lower right would be an example of Durham wheat with differences in straw strength properties. And you can be sure to fit conservation agriculture, the farmers are wanting these strong, strawed, short types. That's what they're really wanting. So some examples of farmer-to-farmer social media. So on the, here's a seed grower speaking about, some of the varieties that he's got for control of the wheat stem sawfly. This is a new variety that came out from Richard Cuthbert at Swift Current. It's a new one with solid stem trait. The seed has just come back from New Zealand, and this farmer has got it. He's got a tote of 1,200 kilograms of seed, and he's all excited. He's hoping to produce about 70,000 kilograms of seed for the fall for registered production. So these are just examples of how the seed, the basic seed, would be put into a tote, a tote of, let's say, about 1,000 kilograms, and then prepared in these specialized facilities and delivered to seed growers for them to do that initial production. Once they get into the second year, it's going to be big trucks, big stuff. This tote stuff is for the very small select foundation stuff. So seed growers know the variety because they grow it. Here is a father-son operators with the new variety Starbuck and Wheatland. And over here on the right is another family farm operators that are processing and cleaning seed that they would have grown on their own farm, and now they are processing it through their various cleaning and grading facilities. And this would be an example of the grain that's been cleaned and tested is put into individual bins of varying size depending on the quantity of the seed that is available. So farmer-to-farmer availability of Pedigreed seed for 2024 as an example. I'm using these just as examples, okay? Just as examples. So this is taken from the SeCan website. So farmers, commercial farmers that are wanting to know what is available, they can look on the SeCan website for varieties. And so if they happen to click on Brandon, they say, okay, it's a Wheat King. It's number 1. It has strong straw FHP. Oh, that looks interesting, but it's protected by PBR and PVP. But there is a technical bulletin here that I could go get a lot more information. And then in terms of availability, each one of these Google Map pins represents a farmer seed grower in his area. And then, of course, SeCan is involved in helping with the promotion of new genetics. And this would be an example of a creative that is done by the SeCan creative team. And another example is Starbuck. And Starbuck was released in 2018. And again, it was just a few hundred kilograms of seed. And the launch was in the fall of 2020. And you can see there's a lot of take-up of this varieties. And the information is it's the best combination of short, strong, straw, disease-resistant yield, 104% of Brandon, MR to FHB, short, strong, straw, all of this information, and then information about intellectual property protection, a mid-stewardship agreement is involved, and plant variety protection in the United States. Technical bullet. So there's lots of information for the farmer to get a hold to see, commercial farmer, looking on the website who is like actually shopping maybe for a variety, a visual of the variety. And then a creative. So, the creatives are, I think, are a great hoot as to the what they could come up with. And here is the starships associated with the Starbuck wheat variety. Another variety, Wheatland, it also was supported for registration in 2018, the same time as Starbuck. And it's a hard red sprig wheat, shortest, strongest straw in image tolerant wheat, 104%, good grade production, intermediate to FHB. Then you get the information about this, the technical bullet. And again, you can see there's a lot of farmers, seed growers, that are expecting this variety to be a good one for sale. And in terms of the creative, we've got this particular ad is a breakout in performance. Now, this is a different example. Ellerslie is another example of a wheat released exactly the same time in 2018, but its properties, its traits are very different. So it's an early 2 days earlier than Brandon. So that's a very big difference, early maturity. So it would be specialized for the northern areas of the parkland area. So even though it was released in 2018, the same time as Starbuck and Wheatland, the expected market uptake of this variety is much less. So the farmers and the seed growers, the seed companies and so on, will be trying to figure out what is the best fit for this new genetics. And the creative. And then we have the farmer availability for another variety called Red Star. And it was released in 2019. So it's a year behind the Starbuck Wheatland and Ellerslie. But you can see that it is, again, one of these varieties that has a very special attribute of being 3 days earlier than Brandon. And it's got a very good disease resistance package and good grade production. But again, it's a specialized variety, so that you can see how the seed growers, seed companies have to figure out the size of the market. So what I wanted to spend a moment on is, okay, in the registration trials, the breeders are bragging about the yield advances of their new genetics. And so the registration trials then serve as a peer review. Do these varieties, in fact, yield? And from the registration trials, they do. But then when you put them into the regional performance trials, what happens? So looking at going back to the 1950s to 2020, in the Manitoba Seed Guide and the Saskatchewan Seed Guide, this is the relative yield, and this has been calculated using a least squares procedure. And you can see that the slope has been increasing, but it has changed. So the rate of genetic gain from 52 to 1990 was 0.34% per year, but then it doubled starting in the early 1900s. And the reason for that doubling was an increased investment by farmers in the genetic enhancement process. So the breeders were able to double their population sizes, apply molecular markers, apply some of the new breeding technologies. So this slide is showing that what is the genetics improvement that's shown up in registration trials is also detected, is also measured, also gathered, also proven in the provincial regional trials. So now this last one is about, okay, what's going on in farmer fields? In farmer fields. So we have yields here going from 1971 all the way to 2020. And this is the yield, the production in tons per hectare. And the data is taken from the Prairie Province spring and canola statistics that is gathered by the provincial governments. So the canola yield is on the bottom side here in the brown, and you can see that it fluctuates with years. And wheat yield is the red line and also fluctuates by year. And you can see that 2002 was a massive drought here. And this, both the wheat and the canola have doubled in that 50-year time period. And which means that the farmers are capturing genetic yield gain. But this on-farm production is not only genetic yield gain, it is better agronomic farm management practices, crop inputs, tillage equipment, conservation agriculture. It is that big suite of agronomics that's going on. So what we see here for this prairie farm production increasing in canola and wheat is a combination of genetic improvement and agronomics. And something that's, I think, is very interesting is that this blue bar is the spread between canola and wheat in 1971. And when we get to about early 2000s, that wheat slope is increasing more rapidly than the canola slope. So the farmer yield gains are being captured by the farmers. So a variety adoption over time, there are many varieties, but only a few dominate. And that's because farmers want better, not more. Most varieties achieve less than 2% of the market area. A major variety might achieve 10% or more, and occasionally a superior variety will achieve more than 30%. Well, I had a video here to show you a changing variety, but I'm running out of time, so we won't be showing you. But the point of that video to show the change of varieties over the course of time is that farmers are always wanting something better, better, better. And the role of the seed grower and the seed companies is to multiply, promote, and distribute the best varieties that are available. So in summary, wheat is not wheat in Canada. There are relative class sizes based on what is the demand for the food products. There are many varieties supported annually, and independent performance trialing helps to sort out which are the best varieties, and this independent performance testing builds farmer trust. The seed grower takes the biggest risk in multiplying seed of the new variety, because while they're not generating that independence performance data, the farmer has committed land to the production of this new variety, and no other variety can be grown on that same piece of land for several years in order to protect the purity of the genetics. So there's trust in independence performance testing. On-farm assessment is critical to uptake. And the farmer-to-farmer dynamics are extensive, and it includes education and support and awareness building of the intellectual property that is also part of, or mid-steward agreements. There's many components of that education and support. So many varieties are available, but only the best rise to the top ranks. And because of that, I cannot overemphasize that it is extremely important for the breeder to exercise rigorous selection pressure so that they are bringing forward the best. and save and if something isn't up there in the top ranks, get rid of it as quick as possible so you can focus your limited resources to find, to combine, to build a better pipeline, to focus, to be sure that you are not bringing forward a me too, but you, in fact, working to bring forward a superior product. So with that, I thank you all, and I look forward to the questions.
Thank you, Ron, for excellent presentation. Actually, there are questions from the audience as well as some of the questions collectively. We link to our own questions. So I would like to relate both of them together because otherwise they would be repetitive. I would like to begin with very good compliment. It came from the Gobzi Chakali. He's saying most of African breeding program relied on yield increment rather than including market demand. So this session is great to most of us. So, and at the same time, he's asking the question, is the process of seed multiplication and commercialization similar to the seed production roadmaps?
Yes. But remember that to go the roadmap from 3 to 13 was a loop done 3 times before it went into commercial production, which was step 14.
Mm-hmm. So I would begin with the question now, which is like collectively gathered from audience, and we make it one. Could you share some insights into recent advancements in wheat research, particularly in the areas such as genetic modification, disease resistance, and nutritional enhancement? So it's like a broad question.
Okay. Yeah. So I'm going to give a couple of examples. When I started as a wheat breeder in the late 1960s, all of the hard red spring wheats were conventional height and might come up to might be 120 centimeters tall. But in order to practice conservation agriculture, the farmers had to manage this straw. So they wanted shorter, stronger straw. So we got the semi-dwarf trait from Simmet, and Simmet got the semi-dwarf trait from Orville Vogel out of Washington, and Orville got it from Japan. The semi-dwarf trait came from Japan after the Second World War. The Japanese had been using the semi-dwarf trait, and then it had been used in Korea. So the semi-dwarf trait eventually made it into North American spring wheats, and we got the trait also in Canada. There was a lot of work that had to be done because the semi-dwarf trait is linked to high yield. And yield and protein are negatively related. So the protein went down as the yield went up. And in hard red spring wheat, you have to have the high protein. So then we had to incorporate the semi-dwarf trait for conservation agriculture. We had to modify the plant through, so that would be a whole nother seminar, so that we changed the plant so that we were able to get better nutrient uptake, water uptake, so that the negative relationship of yield and protein shifted We got higher yield, but the same level of protein, but we shifted the negative relationship. So that's one example of a major genetic modification using a whole suite of traits over time. So that's one. Another example would be the orange wheat blossom midge. It's an insect that damages the kernel. And that trait is a new trait that was introduced in the late or during the 1990s. And it was found in soft red winter wheat, and then moved into Canada Western hard red spring through crossing, the development of molecular markers. We select with molecular markers. The gene has been cloned, and deployed. And so when it comes to deployment, because there is only one gene known so far in the world, it is deployed with an interspersed refuge. And so on the variety Starbuck and Wheatland that you would have seen something about a midge-tolerant stewardship agreement is in place. So those are just two examples. And there's examples like we've been practicing marker-assisted selection going back to the 1990s, double haploids.
So moving to the next question, with climate change increasingly affecting agriculture practices, how do you see the future of wheat farming evolving to adapt to these challenges? So I would like to also merge the questions. Dr. Atul Kumar Panchuri, Assistant Professor, India, he's saying, how do we leverage existing genetic diversity to create a climate resilient crop? Because both are climate change and climate resilient crop. So you can merge the answer, so it would be easy.
Yeah. I think, first of all, one has to have an understanding of what is the climate and the climate projections, because they might not be the same in every region. Now, there are some very good global models, but I would say that's number one. Have a look, see what is the projections for the climate in your area, because there might be things that can be done about planting earlier or planting later to be in better synchrony with what are going to be the temperatures and the rainfall distribution. It might be that there could be a shift as the winters might get milder. You might be able to grow winter wheat instead of spring wheat. So there's various things that can be done with regard to understanding the climate and then fitting the genetics, the crops, to what is the projected climate. In terms of building using genetic resources, that becomes the big challenge of finding out what is the key traits that one might be needing. Like, for example, let's say that the projected changes are that there is going to be more likely temperature spikes, high spikes up into the 30s, high 30s, during critical flowering time. The wheat is most sensitive during the flowering period. And when and not just the flowering period, but when the flowers are being initiated, and the wheat plant goes reproductive, changes from vegetative to reproductive at the 3-leaf stage. At the 3-leaf stage. So when it is in that early stage and you get heat shocks when it's going reproductive, and the spikelets are developing, pollen is developing as you're going up into the booting stage and so on, it's very sensitive. And some varieties are very sensitive during that time period. So in terms of genetic resources, that would be an example of looking for genetic resources for that trait. The incorporation of the trait into an elite variety is a whole nother complicated step.
That's really great. I mean, it's a like two ways process. Like you need to make different plans, A, B, C, D, and then you have to parallelly go for to develop the climate resilient wheat or any
Yes,
crop. Yeah.
yes, exactly. And you might have a trait that's got better tolerance to a pre-anthesis heat shock. But if it's put into a variety that is weak strawed, the farmer isn't going to want it. Or if it's susceptible to disease, or it's, sold at a low price. So it takes it takes a very big strategy to move that trait into an elite background that a farmer will grow. So you'd better really be working on your strategy. You've got a genetic resource, you have the way by which to track it as you're incorporating it. Can you keep it? Can you follow it? And can you hang on to the big bag of other traits that you need?
Nice. So moving to next question, again, I would merge with the audience question. So the question is, speed breeding technology has gained traction in recent years for accelerating the breeding process. What are your thoughts on its potential to revolutionize wheat breeding and crop improvement efforts? And this question comes from Satyapal Singh, corporate professional. He's saying, is it possible to increase the genetic gain through speed breeding? So both are for speed
Yes,
breeding.
yes, okay. So speed breeding involves multiple generations per year. That's the core of it. And as you increase your speed, your cycle, you have to be aware of breaking the seed dormancy between generations. If you don't break the dormancy, then what you were doing is selecting varieties that will have less dormancy, no dormancy. So yes, it'll work in a speed breeding situation. You can generate the cycles very rapidly, but the end product might be that it doesn't have dormancy. And when you get that kind of a variety into farmer fields and it sprouts and the functional quality goes down, there is a huge loss. So when you do speed breeding, you have to do something about breaking dormancy. And if people want to know about breaking dormancy, I wrote a paper on breaking dormancy I think I wrote it back in about 1976, 77, because we were wanting to accelerate generations in the greenhouse. And you have to we had to break dormancy and yet retain the dormancy, because in our environment, pre-harvest sprouting is a big issue. So speed breeding, yes, but you got to be sure you're not getting some sort of unintended consequences. The other thing about speed breeding is you rapidly accelerate the generations to get an inbred line. And if you will look at the presentation that I gave on May the 1st, I made the case that your highest gene frequency of the desirable genes is occurring in the F2. And it achieves equilibrium when you've got inbred lines. So that is a negative. So you've got to be figuring out, how do I balance the pros and cons of accelerated generation advancement when I don't have as many opportunities to select for the desirable genes. So you have to really figure out how you're doing these things and why. Think it through extremely well.
So definitely genetic gain, if you are accelerating the generation, so it would affect the genetic gain through speed breeding.
Yeah, well, you got to be careful. Yes, you got to be careful. You've accelerated, you reduced the time, but you might have made a very poor pre-harvest sprouting susceptible line. So you haven't accelerated genetic gain. You've made a problem. But by the equation, yes.
Yes.
The other thing about that breeder equation on accelerating genetic gain, reducing the time between breeding cycles, is your choice of parents might not have been tested adequately. So you've got more errors in the phenotypic information about your parents. So yes, you have reduced the time between crossing cycles, but you might have a higher or poorer choice of parents because your phenotypic information isn't so good. So time between crossing generations is not the only factor in that breeder equation on genetic gain.
Yes, that's true. So moving to next question, as we look ahead to the future of farming, what role do you envision for next generation farmers? And how can we ensure that they are equipped with knowledge and tools to navigate the complexities of modern agriculture?
Yes, that's an excellent point. And I look upon it as the professionalization of agriculture, the professionalization of farming. The farming tools, like no matter where you are around the world, there are cell phones, there is internet. And this is a great advantage for the next generation of farmers to be able to explore and use some of the new ways of learning, getting information about products, getting independent advice. Yeah. And I think of it as, this continuous learning and adopting. Yes. Encourage the young ones, and even in their crazy ideas, to put a robot on a bicycle.
So leading to next question, because we have so many information and so many resources these days, so time management is a big issue. So time management is crucial for successful research and farming operations. What strategies or practices do you employ to optimize your time and productivity in your work?
Well, right now I'm retired and I'm a science advisor to SeCan. But what I did as a scientist, I had a very, very, very clear goal for my breeding program. And that was the focus. That was the focus. Reports could be done at a little different time. However, I also did not work an 8-hour day or a 7.5-hour day. I worked many long hours, and my spouse was extremely supportive of my spending time on genetic enhancement. But focusing was extremely important. And when having conversations with people, I tried to make it as to the point as possible to be, saving time. And when it came to be giving instructions to facilitate good instructions with the staff who are going to be doing things, I would write out the work order, write it out, and then with the lead technician, discuss it. Was it understood? To be sure that it was implemented according to what I thought. Whereas when you do just a verbal communication, all kinds of things can go wrong. And that then becomes a big waste of time. So focus, a plan of communication, written communication, oral communication was extremely important.
So focus is a key. And that leads to my personal question. Being in science, everybody says that work-life balance, like as you said, your spouse was very supportive and you need to devote a lot of time in science, not only those working hours, 9 to 5 job it is. It is like more than that, because you need to write articles and so many other things back of your mind. Even if you are available for your family, you back of your mind is thinking about the research and science. So what is your take in that?
Yeah, a work-life balance. How does one work at it? How does one define it? So for myself and my spouse, we have established, let's call them rituals, so that that in the ritual we focus completely on one another. There's time apart. There's time together. When we're together, it is extremely, extremely focused. Yeah, I was extremely fortunate to have a spouse that knew of my passion and supported me in my passion. And she said, you can pursue your passion in genetic enhancement as long as you do not go into management.
Nice. As you said, the second thing, communication after focus. So that works. So moving to next question, ethics in research and agriculture, seeds and variety are increasingly important topics. How do you approach ethical consideration in your work?
I didn't, please repeat it, Priyanka.
So it's like a ethical consideration at your work. So ethics in research and agriculture, especially for, seed and varieties, are increasingly important. So how do you approach ethics?
Ethics, yes. Okay. Yes. Integrity. Yes,
Yes.
ethics. I would say that should be foundational. When you are out scoring for disease resistance. I'm going to go back even further. Making a field book. The field book has to be correct. The randomization has to be correct. The printout of it, the planting of the experiments has to be correct. If there is any error, we're going to have a problem. When you are scoring the diseases, that you score it as per the criteria. You can't say, oh, but this is my favorite line, I'll give it a better score. You can't do that. You score it as you see it. And everything is scored as you see it. Everything has got to be done in an ethical manner. And by that, I mean, you're following good lab procedures. You're following good field practices. And then the data is the data. You look at the data, you analyze it. Are there outliers? Can you explain things? Yeah. Ethics in genetic enhancement, I say, is foundational. It should be in everything, but it's foundational. And I think of ethics as just correct behavior.
So definitely, so as you suggested, it begin with your field book. I mean, prior to when you plan your randomization to field book recording data, so it's begin from the very core.
Yes, yeah.
So last question, not from the audience one, the one which we collected from all of the audience. So collaboration is often key to driving innovation and progress in agriculture. Can you share any examples of successful collaborative projects you have been involved in and the benefit they have brought to the field of beet science?
Okay, there's several examples. One is when I was speaking on the May 1 lecture about the environments, the selecting environments, that the selecting environments spanned from Lethbridge in the west to Winnipeg in the east, a distance of about 1,200 kilometers. So that involved then collaborating with scientists in the Lethbridge area and these other areas all the way over to the Winnipeg area. So you have to have collaborations with those particular people. You have to have funding for them to do the work. You have to help to get the funding for the work. And then when if you succeeded getting something, then there would be the sharing of the research credits about that. That's one example for agronomic testing, the disease testing, and all of that. I'll give you another example. A fellow by the name of Julian Thomas wanted to incorporate 4 different bunt genes into 5 different backgrounds. And he got farmer funding from the Western Grains Research Foundation, and he put that material, did all of the back crossing, and then he wanted to go into the field and test it. So we contacted my colleague, Ron Knox at Swiftburn, and Julian Thomas was in Lethbridge, way over that west side. And he contacted Ron Knox and myself to do the evaluation, to do the field evaluation for the bunt reaction. And then because they were put in these bunt genes were put in different market classes, we wanted to see, okay, could any of these be deployed into a particular market class? So then we agreed if something came out in this area, who would be the lead author? If something came out in that area, who would be the lead author? So a variety came out by the name of AC Foremost out of that. It got the BT10 gene, AC Foremost, very widely grown as a Canada prairie springweed. I shouldn't say widely grown in a high productivity area of central Alberta. And it was released there in, I think, about 1996. And it's been very hard to knock it off because it has some very unique strong straw attributes. And then in the hard red springwheat background, the BT10 gene was released in a variety called AC Cadillac. And I was the lead author on that one. But unbeknownst to us, when Julian Thomas arranged for the testing of AC Cadillac with that BT10 gene in Njoro, Kenya, it showed to have resistance to the UG99 strain of stem rust. So the SRCAD gene comes out of a collaborative project for bunt resistance. Unbeknownst to us, tightly linked is another stem rust resistance gene that is, and also in in AC Cadillac is LR34. So the LR34 helper gene with the SRCAD gives an MR to an R stem rust reaction. And that's an example of a collaborative project that has been shared around the world. And yeah, they've got, yeah. So those are 2 examples.
Sorry. Now I would like to move to the audience questions. So the first question from the Grace Gland Gavisai. He is asking what wheat varieties are suitable for North American and Canadian market, and what traits do growers prioritize when selecting them? What is your vision for the agriculture sector? Oh my God, there are multiple questions. If you could live twice, how would you double your impact on the world and reduce hunger in certain regions? Oh, very good question.
Okay, let's go to the first one. What was it? Canadian
and North American market traits which grower prioritize.
Okay, so that's I would say is too general. It's too general.
Yeah.
So I would give you an example of of uh, of Western Canada. Um, a variety that was released in 2012. So multiplied in 12, 13, and 14. So it went out to farmers for commercial launch in 15. The farmers liked it so much that it became #1 variety in 2016 and continued to be the top variety in 2023. So what is it that farmers are seeing in that variety to make it a top variety? So that's a good example, eh? It's a top variety for 8 years or 7 years, whatever it is. It's from 2016 to 2023. 8 years. So #1, it has a high yield. High yield. No loss in protein. It has very good disease resistance, including an MR rating to FHB. It is eligible for the top grades of hard red spring wheat. And it is on a strong strawed semi-dwarf stature, so that farmers can put inputs like uh, fertilizer, and it'll still stand up. It has broad adaptation. So when it is good growing conditions, it responds. When it is dry conditions, it still is responding better than others. So it's got very good water use efficiency and nitrogen use efficiency. So you can see it's a massive bag of traits that are in that particular variety. And then the other ones that I showed in the presentation, AAC Starbuck and AAC Wheatland, it looked like there are improvements in yield over Brandon, and they are on a short straw stature. And Starbuck is MR to FHB, Wheatland is I, so that there's complementary traits there. But the farmers, seed growers and farmers seem to be really adopting them. So that's what it comes to about when there's a new variety that becomes big on the market and stays there, it has surpassed the others. And then you use that one as an elite variety to put in other traits to go the next level, because you only make advance through transgressive segregation.
So in one line, we can say that it is a broad adaptation. We would look for many, many traits, not only like some few traits.
Yeah,
so,
yeah. It's a
so
big bag.
I don't know if you would like to answer this. If you could live twice, how would you double your impact on the world?
I would have to find my spouse sooner.
That's good one. So moving to next question, it's from V. Rajendran, PhD student. How is the genetic gain determined in crop breeding program, and should genetic selection be integrated with the existing breeding platform to increase the without expanding program size? So
repeat the question.
Pretty quick. How is the genetic gain determined in crop breeding program? So I think you already answered many times this. And should genetic selection be integrated with existing breeding platform to increase it without expanding program size?
Yes, I agree with genomic selection. I agree very much with it. But I think one has to proof it out in one's gene pool and target environments. And I would say, don't rely on genetic selection using a single location as your basis. Because if you're wanting broad adaptation, you'd better be doing testing your training population over a broad range. I think early generation selection at single locations has been a major limitation in the rates of genetic gain.
So moving to next one, Dr. Muhammad Imran, assistant professor, he's asking, what is the current breeding status of black wheat in India and worldwide? How to plan effective breeding strategies for black wheat?
Black wheat. What is black wheat? Is that the chaff or the color of the grain? Or is it blast? B-L-A-S-T.
He said black. So I think it's the chaff, because we in general, we see on the, no idea. So we can
Yeah.
skip this question. So moving to the next one, does enhancing the average genetic value or the average phenotypic value through selection within population contribute more significantly to genetic gain? So average phenotypic value through selection, it can contribute for more significantly to genetic gain?
Yeah, I would say it depends on what is the correlation between the phenotypic expression and the genes. If there is, the expression of the phenotype is primarily due to the genotype, then yes, you'll make good gains. But remember, the phenotype is an interplay of the environment and the genetics. So you want to be sure that when you're selecting the phenotype, that it is due more primarily to the genes than the environment. I'll give you an example. Let's say you're selecting for rust resistance, and you're selecting in a nursery for those phenotypes that show no rust. And then you take your phenotypic type into another year, put it in the same nursery, and those that were resistant are now susceptible. The reason being that the year before there was either no inoculum or the conditions weren't right. So you have to remember that your phenotype is the interplay of the environment and the genes. So you want to use your environments to be as effective as possible to get the genetic expression. My computer is saying the battery is low, and I don't see why that should be. Anyhow, let's keep going.
Yeah. Okay. So I would like to ask Subo, can you please come and because most of the questions which you collected right now
Is
are
covered.
from like we already answered in a way that I don't think so we need to repeat. I don't see many of them are useful. Actually, they are asking what is the meaning of the CWS. I mean, the term, the acronym which we use for Canadian wheat spring.
Yes.
So those
And
kind of.
I just messaged you a variety name. Priyanka, you can Google it. It's one of our viewers is mentioned. He worked on that wheat. So black wheat actually the color of the wheat. I think any anthocyanin or something is different here. So it's look like a black color. Like
Yes. Yes.
I just name. Yes. So maybe it is some continent, some area, Pacific area is there.
Yes.
I
Yes.
have a personal question with you. Like what I understand from your presentation that farmers are play a very big role there. Means they have a own field, they have a own social network group, and they have a connections. But
Yes.
for India and developing countries, the small holders, farmers are mostly a small holders, and uh, and their varieties are very different. Means in a small plot you can find multiple varieties. No one knows which one is better. Okay, and they are moved with to the demand of the market in another way. Means if the price is high a particular variety, next year everybody grown that particular variety and forget about everything. So here the culture is little bit different. So I wanted to know that if someone wanted to exchange their material, like you did, there is some specific process. But here, if one state farmer is wanted to share their material to another state, then the information is not so concrete. Means it is the personal experience of a farmer, not so concrete informations are available. So my interest is that is there in Canada for such type of situations when intervene is required between the farmer and a scientist, or a farmer or a institute where they can catalog the things and share the information in right way. So that is, I think, is a missing. Sometimes it's there, but not actually practically available. So what we can do for that thing means how we can educated farmer, or how we can learn them that okay, these are the characters. This is not for this area. It is for only that area and that season. So these are some missing link, I can say. I am sure this is government wise it be there, but in practically it's not visible so so importantly or so significantly. So what is your suggestion for such country where the farmer education is not up to mark, or they are growing catching up and field is very small, they don't need, they don't understand the actual importance of variety and why it is made for them. So what we can do?
Yeah. Yeah. Like those are very, very good questions. And that's where I think farmer to farmer interactions are very important. But also there needs to be like, where is let's say, the most respected farmer? And does that most respected farmer have good standing in the community for his knowledge and his integrity, and because they become important in helping to move information. And then another part of this is, are there any opportunities to have like small demonstration plots of something new with something that is traditional? Like let's say a leading farmer, can he be somehow drawn into a network of where the new genetics is on his farm, where he can see it together with his current genetics? Like farmers are very influential with one another. And getting independent information is a huge challenge. It's a huge challenge. But once a network is set up and functioning, it serves good purposes. I know. Remember when I was in Kenya in 1970, 71, 72. We did demonstration plots. We would do on-farm field trials. We'd arrange with farmers that we could get land from them to grow field trials. As a matter of fact, I was with our team planting at one of the locations in Kenya, and a car stopped and was called over. And there it was, President Jomo Kenyatta saying, "Hey, what are you doing here? This is my farm." So it was really a wonderful opportunity to explain what it is that we were doing. And of course, he would be an example, somebody overly influential.
Yeah.
But yeah.
Yeah. Uh,
But
Priyanka,
but yeah, just to me, Shubha, I think it is extremely complicated on getting new genetics, new technology into farmers' hands. It is a big, big challenge and something that we have to keep on working on. But I think leading farmers can play an even bigger role.
and maybe internet will be helpful for this, because in India, Pakistan, Bangladesh, Philippines, we have a very low cost internet. So
Yeah.
maybe the networking now it's better. Maybe the connections will be better. And
Yes.
interestingly, last few days, I observed that in BioEngine, many people are registered. Those are farmers. Means they are putting their occupations as a farmers. So maybe
Ah,
it's an interesting things that yes, because ultimately farmers will do actual work. We are starting, and our work is limited in a certain way, but they are the actual who can carry forward this thing. So yes, your and what that is, I just recall your ethics part. You are covered the research ethics well, but what happened here, we are very worried about giving the seed to others. In case of developing countries, or you can say in India, Bangladesh, we are very worried when we share the seeds, because of it may be hijacked, it may be misused, it may be someone is misused in other ways. So yes, and that's why I think we are also focused on ethics part, that it's need to be people need to be serious and educated about things. And yes, your point of view is a very correct. If the taking data is wrong, then the everything is wrong. Means if someone not going to the field and putting the data in from outside of the field, then is very difficult for that. And
yeah.
I just it may be last question from my side. Just we are sometime we are, because we are mainly rice eating people. Means most not such wheat people. Those in India it's a 50-50 scenario. But gluten in free gluten free wheat is just a trade name, or it's a serious issue in Canada? Means people are avoiding it, or is just a marketing strategy?
Okay. I'm, I'll get to it. But first of all, I want to say uh, that participatory plant breeding, which people talk a lot about, but I would say that when it comes to um, variety adoption,
Hmm.
the farmers are participating. They are growing it on their fields, they're learning about it, and they will say, yes, will work, or no, it will not work. So there's in this procedure that we have in Canada, this I think is a very good example of participatory genetic enhancement. They're not involved in the crossing, but when it comes to that final step of will it work, and all of these different areas, they have a big influence. The other thing I wanted to say, Shubho, is with regard to ethics, is that with regard to seed, the material transfer agreements are super important.
Yes.
And also respect for intellectual property. You know, like if something is got protected by plant breeders' rights, the farmers have to respect that as well. And the seed companies are involved in, enforcement of plant breeders' rights. But yes, uh, um, uh, there's a lot of ethics that has to be done around the genetic resources and material transfer agreement. And then to go to your last question about gluten intolerance. Gluten intolerance is very real. Okay, some people really have a gluten intolerance. That's the way it is. You know, some people have a peanut intolerance. Some have a lactose intolerance. This is just part of the human genetic makeup. When it comes to what makes the elastic properties of wheat so that you can make a leaven loaf of bread, to make a leaven loaf of bread, then it is the gliadin and glutenins that come together, they're hydrated, and that makes gluten. And when the gliadins and glutens are coming together and are hydrated, they have elastic properties. So when the yeast is giving off carbon dioxide, you get a cell. And if it's got good enough gluten strength, that cell will remain intact so that you get the leavening. So if you want to have leavened bread, it's going to be pretty tough to get rid of the gliadins. But you can get rid of the gliadins and have other kinds of products that don't require that elasticity. But if somebody is a real gluten celiac, then you can't take any risk of having even one kernel of a contaminant in there. So, I think gliadin-free bread or gliadin-free wheat is something that would make a product for certain kinds, certain specialized products.
And very few percent market share out there, I think.
Yeah.
Okay. So Priyanka, I think it's enough. So many questions, and we enjoyed both your webinar. We are very enjoyed, and hopefully in the future also. It's a great pleasure to meet you and learn many things. And last day we also talked about, and today is also Soma also, me and Soma, and like Priyanka, is uh, life partner support is very important. What we understand, and communications is one thing. Sometimes we missed it. Sometimes we are. Sometime we need some more time, but due to miscommunications, we can't understand each other. So yes, it is a very good takeaway messages from you that partners is important, and also the communications with the partner is very important to manage our work. And yes, we are researchers, so our it's not a, 10 to 5 p.m. job. So sometimes it's delay and sometimes it's early. So yes, we consider and we try to focus your message in our life also. So it's a great help for us, and lots of appreciations we received. And as you know, many diverse people are attending, from students to professors. So they all are get something from you, and they are very happy for your uh, lecture delivery, your presentations, and your depth knowledge. Mainly today, many people are talking about your knowledge in this field. So thanks for coming, BioEngine, and we will keep in touch with you obviously Priyanka will be there. And when we need any guidance, any suggestions from you, we will back to you, and hopefully you will there for us.
Yes, yes. I would very much, I've really enjoyed this very much, and it's, yeah, I enjoy sharing, yeah. And if there's other lectures you think that I might be able to do, I'm most willing to see about preparing and
Yes.
sharing. Yeah, this has been
Yes.
a real pleasure for me to connect, and then to think that you've got this massive audience. Yeah. Anyhow, I really appreciate this.
Thanks. Thank you.
Thank you so much, Ron. I would really appreciate, and we are highly honored to have you on BioEngine to share your life experiences, its broad experiences, and also the knowledge, which is for all of us. And we all are learning from you and from your life. So thank you so much.
Yes. And for the audience, I already shared the feedback link in the in the chat box, and I'll share once again during when I close this live session. Our website is down from yesterday night. I'm trying to recover it as soon as possible. Already mailed and contact our service provider. I don't know what happened, but maybe tomorrow it will works again. So don't worry about the website. Everything is in your emails, and we will give the participant certificate after 4 or 5 days, because we have another webinar in 18 on the AI in writing and manage and the searching of articles. So after that webinar, I will have some free time. Then I prepare the all certificates. So after for 4 or 5 days, you will get the certificate. In the meantime, website will be the recovered. And see you all of you in next webinar. And once again, Ron, thank you,
Thank
and namaste. Thanks,
Yeah,
and
yeah, yeah.
thanks very.
Thank you
And
all. Yeah. Bye-bye.
thank you.
bye
Thank you.
bye,
Bye.
bye bye.
Yeah,
And
bye.
Priyanka, thanks to arranging this webinar and helping me out. See you
Thank
one
you.
again.
Bye.
Okay, bye.